Okay, hey, biology students from Wyzetta. This is Mr. Kemmis again. We're going to do the dihybrid cross notes here. So I got a couple tricks to show you today. And by the end of this video, you should be able to do dihybrid crosses. So I'm going to open up the notes in Notability so I can draw on them. This should be able to help with you. So I'm going to share my screen so you can see that. There we go. All right, now you should be able to see my screen here. If you're looking for the notes, this is it, 01B, dihybrid crosses, that's in the resources folder there. So dihybrid means two traits. So we're gonna go ahead and do the two traits at one time. Do you think we can keep track of that at the same time? Thank you, Notability. So di means two, and we're gonna go ahead and do two traits at the same time. Now we do need to remember the law of independent assortment. These traits are gonna move independently of each other. They're not gonna be linked. This is gonna be the bigger Punnett squares, the 4x4 size. It should be a little bit bigger. Okay, so last notes, we looked at the different steps for solving Punnett squares, and you're gonna wanna use this same one here. Code the problem, figure out what do the letters mean, what is dominant and what is recessive. And then go back to the word problem, figure out the genotypes of the parents. Sometimes that'll be given to, sometimes it won't. Next step is to determine the possible gametes. This is a little bit harder for dihybrid crossing. I'll show you a couple ways to do that. Step 4 is also kind of difficult here at the 10th grade level for dihybrid cross. You need to keep track of the letters. I'm going to teach you a little bit how to do that. And then step 5, report the results or answer the questions that are assigned to you there. So let's look at this first one. Why don't we go back to Mendel's garden and look at his peas. We have round is going to be dominant to wrinkled, so I'm going to use a big R and little r. And notice this is step 1 here for coding. Big Y is going to be dominant to little y. We're going to have the yellow and the green below there as well. Okay, so this is we're going to have to use this to determine the gametes. We can use these letters. We can figure out the eggs and sperm here in step 3. All right, so let's do a couple practice problems here. So this is all step 2, just figure out, could we do homozygous round and heterozygous for yellow? What would that parent look like? If we just had the words, could we figure out what are the letters? So homozygous round, okay, look, big R is gonna be round. And homozygous is going to mean the same. So I'm going to use big R, big R. And then heterozygous for yellow. Hetero means different. So I'm going to have one capital and one lower. Make sure you exaggerate your capital and lowercase here. And then we can check our answer. I figured out how to use these sticky notes earlier today. It's kind of fun. You can check your answer. Did we get that one right? Big R, big Y, little y. That's going to be parent number 1 if we were going to do a problem set here. All right, parent number 2. We have heterozygous for round and also heterozygous for yellow. They don't have a word there for yellow, so you're going to want to assume that's heterozygous as well. So how would we do double heterozygous? We're going to have big R, little r, and big Y, little y. Sometimes I use cursive for the second letter if that helps. And so then we can check with the sticky note. How does that look? Move that down. Oh, you can do it. I believe in you. There it goes. And you can see, hey, we have big R, little, big Y, little y. Do you see how that works? You want to try another one? You could try those at home, and then you can pause the video now, try the next 3 at home, and then hit play again. We'll reveal the sticky note. All right, I'm glad you're back. Here's the correct answers for those next 3. All right, so now that we know how to code the problem and write down the genotypes of the parents using the vocabulary, we can go ahead and start to determine the gametes for each parent. This is the trickiest step, but we're going to pull in a math test here, so you can try this one. We're going to use the FOIL method in dihybrid crosses to figure out the gametes. Okay, so here it is, determining gametes. This will be step 3. So we're going to use the FOIL method. And if you recall from math, the FOIL method is first, outside, inside, and last. So for this one, I'm going to take the first R and the first Y. Look, they're both capitals. And that's going to be my first gamete. So that's going to go into a sperm or an egg. Then we're going to use the outside. I'm going to use different colors here. The outside letters, big R, little y, that's going to be the second gamete. And then the inside letters, little r, big Y. We want to have one letter for each gene here, one R and one Y. We're just trying to figure out which one. And then the last letters is going to be the last R and the last Y. So that's going to be a double recessive, little r, little y. So if that was our first parent, that would be what would go down the side or the top of your Punnett square. You can see, oh look, I got them all right, way to go. And then parent 2 has the same genes here. So a shortcut is they're going to have the same gametes, or you could try the FOIL method again at home. So let's see, did we get that one right? It's going to be the same for parent number 2. This is a typical Punnett square cross that you would see, is that both parents are heterozygous for each trait. All right, so let's go to the next square. Next step. Step 4 is to do the cross. So you need to set up a Punnett square. And so here would be, for example, parent 1. Here would be parent 2. It usually does not matter if you put parent 1 across the side or across the top, but it does matter that you put the letters in order the way that you FOILed them. So make sure you do that. Notice big R, big Y was first. That's going in the first box. Big R, little y, the orange one was second. It's going to go here. And then here's the third gamete and the fourth. And then repeat that across the top for the second parent. It's the first, second, third, and fourth gamete. Okay. So now it's just about filling in the letters. This one's not too bad, but it is quite a bit of bookkeeping. If you have bad handwriting or you rush, you're going to make some mistakes here. So please be careful with this one. And I'm just going to go ahead and reveal a little bit here. Okay, here's the first row. So for this one, I take the big R and combine them. I'm just going to draw on here. Keep your R's together, okay? And keep your Y's together as well. And just fill those in, okay? You always want to go, I usually go in alphabetical order. That helps me. Some problems might not be in alphabetical order, and so then you could make sure you always have the same letter first. And then keep your R's together and keep your Y's together. All right, if we go to the second box, you can see how we fill those in. Which green did I have? Oh, whatever. This big R and this big R are just going to go here, so we have 2 capital R's. And then for yellow, we're going to have a big Y and a little y. And then you could keep going here. Big R, little r, and 2 capital Y's. Big R, little r, capital Y, lowercase y there at the end. So that would be your first row there. And then you're going to want to keep going and fill out the different rows and columns here until you have all 16. Another mistake here is you don't make a big enough box for you to write 4 letters. Make sure your boxes are big enough on your Punnett squares when you're trying it at home. So let's look at the second group here. We're going to start to see some green, some double recessive. Those 2 little Y's are recessive. Maybe some green peas. Okay, row 3, we're going to start to see some wrinkled as well. Some little r is going to show a wrinkled P. So we'll see those. And then here in the last row, you would start to see some more patterns here. So that's step 4, get all your letters in here. Now this one's kind of nice because it actually shows you the phenotype as well. Okay, that is the round or wrinkled, the yellow or green. So it's kind of a little bit of a helper for us as we're starting to get to know these. So you can find the genotypes here in the letters. The phenotype is what do they physically look like, what color, and smooth or wrinkled for the P's. Step 5 is an interesting ratio. These are kind of tough. Usually we just look at the phenotype ratio, okay? And a common pattern here is you're going to want to see a 9 to 3 to 3 to 1. So the first one is the one that's dominant for both traits. So you'll see those here. They're all yellow and smooth. Okay. The next 3 you want to see here are going to be green and smooth, or round and green. Here you go. That's going to be those here. It's going to be green and smooth. Our next color we'll have usually right here. So that's going to be recessive for your first trait and dominant phenotype for the second trait. So that's going to be yellow and wrinkled. That's going to be that third 3, or that second 3 there, the third number in the ratio. It's going to be yellow and wrinkled. And for our last one, we always see this down here, this double recessive in the lower right. Okay, look for that. It's recessive for both traits, so it's going to be green and wrinkled. I'm just going to pop that over here so it fits. Green and wrinkled. There you go. So look for that pattern. You should definitely see that one in dihybrid crosses, the 9 to 3 to 3 to 1. These will also be in the same places for where they're dominant or recessive for the phenotypes. So look for that. All right, let's keep going here. Let's try another one with Squidward. Anybody watch SpongeBob or something? So I already set up the color and the size of their nose are the 2 traits that we're going to test here for the dihybrid cross. So blue is going to be dominant to green, and then large nose is going to be dominant to small nose. So let's look at Squidward's and his wife here. Squidward is going to be heterozygous for body color. He's going to have 1 dominant blue allele and 1 recessive green. And then his wife is going to be green. So she's going to have 2 recessive alleles, little b. And then let's look at their noses, okay, shall we? Squidward's nose is a little bit larger. It's hard to tell in the picture there, but they both have large nose genotypes, big N, little n. They're going to be heterozygous for nose types. So you could go ahead and do the FOIL method here. I'm going to do it. I'm going to do it for Squidward's wife. How about, should we do FOIL for his wife? For Squidward himself, it should be similar to what we just did. And so I don't think there's any tricks there. All right, so for the wife's genotype, the first letter is little b, big N. The outside letters are going to be little b, little n. Inside letters, little b, capital N. And the last letter, the last b and the last N, little b, little n. So here you can see she only has 2 types of genotypes, okay? Little b, big N, and little b, little n. Squidward himself is going to have 4 possible gametes there. Another way to do this is to make a little Punnett square. Sometimes this confuses kids because it's, why are we doing a square before doing the actual cross? But this is another way. You would put the B's on one side, the N's on the other, and you could figure out the gametes. So I have seen that in some other videos, and I know some other teachers do use that one. If that makes sense to you, great. If you're like, chemists, look, FOIL method's good, then stick with that. All right, so let's go to step 4. We're actually going to do the Punnett square. Okay, so if just looking at these, we can kind of figure out where the wife is. Here's the wife down the side here. There's her genotypes of her gametes, her possible gametes. And then here's Squidward himself. you could see his gametes down here. Now here's a shortcut for you guys playing at home. Look at the first two rows and the last two rows. They're the same. So really, we can figure out these ratios by skipping those last two rows because it's gonna be equivalent. So I would recommend to my students just doing the first two rows. That'll get you the answer. Okay, we can skip that a little bit and simplify things. So here we have their genotypes all filled in. Okay, big B, little b, big N goes here. The second box, we had big B, little b. Make sure you have that capital first and the B's together. Big N, little n. The third box, two little b's, one from mom and one from dad, and two capital N's, one from each parent. And then here, the fourth box, little b, big N, little. All right, let's go to the second row. Big B, big N, little n. Make sure you keep the B's together and the N's together. Capitals always go first. Big B, little n, little n. Little b, big N, little. And then, oh, surprise, in that lower right corner, we have the double recessive again. Little b, little n, little n. All right, so that's step 4, do the cross. If you want, you could still go through and do this last third and fourth row, but again, we're gonna get the same answer. So there, okay. All right, now for step 5, it's asking us, we don't even need to find all of the genotypes, but just what fraction of the genotypes would have green with small noses? So let's go back to our coding. What is the letter for green? So if we look here, green is little b. So what's the only way we can have little b's? You're gonna have to have, since it's a recessive trait, you're gonna have to have 2 little b's. So this question is asking us, how many of the children have little b? And then small noses. Do you remember, was that dominant or recessive? Let's go check. Small noses, here we are. Small noses, that was recessive. What's the only way to get a small nose? You need a little n from mom and a little n from dad. You need to be homozygous recessive. So it's asking us in this question, here's small noses. How many of the children are predicted to have 2 little b's and 2 little n's? And if we go up to our table here, we can see that. Is this one? No. Just that one in the lower right. Do you see any more? So what's our fraction then? 1 out of 8 boxes here would be right there. And then we can go down and we can see our options. These options really don't help unless you do the work first, okay? Okay, a question from the audience. What if we went and did this whole chart? Would we still get the answer 1 out of 8? Well, let's see here, shall we? Where's the double recessive? We have 1 here and 1 here. So our fraction would be 2. And how many total boxes of children do we have? 4, 8, 12, 16. Could we reduce 2 16ths to anything? Well, yeah, let's reduce that. We get 1/8, and that's going to be the same answer here. Some teachers will have you express these as a fraction. Some teachers will have you do this as a percentage. Just look at the problem and see what they are. But that's pretty much about it for that. All right, well, that has been your dihybrid cross. You should be able to solve problems using those steps. Let's stop sharing the screen there. Go. And then you should be able to do page 4 in your homework number 1. That one is going to be due on Wednesday. So thanks for watching. If you have any questions, send us an email, drop a comment, like, subscribe, whatever you want to do there. And we're going to stop recording. Have a good day, Wayzata.